now we will move to the COVID-19 and the participants, the COVID-19 theme and the participation of water scientists. Actually, when I was thinking how to make, what should be the name, I didn't come up with some of us, of course, Dr. Kitajima or Dr. Arun, who is already are there, or even the Dr. Honda. Of course, many of us were knowing the virus and water, but many has rose to the occasion. Like I certainly, in my case, I and several, we rose to the occasion and try to understand that what is going on in how can we participate in COVID pandemic and like that. So this panel is had a lot of participants, and even I was mentioned myself there, but then I removed myself because I wanted to listen these for I think I will say the very, very good worker. Actually, not only they work, but they also spread the, so I don't know that this is Professor Senon, Senon Bartlett, Professor Honda, and I will introduce each of them. Professor Kitajima and Professor Dr. Arun is here with us to talk on the climate change. Professor Senon, okay? So we would go with like, I try to arrange the, so in that case, let's invite the first, of course, Professor Senon is still on the way, I guess. So let's, can I start with Professor Honda if he permits, because he is my very dear friend. So I can ask Professor Honda to start. So how we will go is that we all panelists will be talking 5 to 7 minutes, 7 to 10 minutes. You can take and you can talk about whatever you want on the COVID-19, that what was your experience, what are the challenges you are taking now or seeing now. So whatever the message or with or without PowerPoint. So let me introduce the Dr. Honda, and then we will move to some questions that I have prepared, which is not so difficult, just about that, what we should go ahead. So Dr. Ryo Honda is a graduate from Todai and the University of Tokyo, and he has established himself as a teacher, as a researcher in a big way, and now he's working as an associate professor at Kanazawa University. He has received the WET Excellent Paper Award from Japanese Society of Water Environment. Actually, when I was doing PhD there, and if any of our mates receive any award from this very auspicious society, we used to go for party. So that is really a good society to get recognition from. Dr. Honda is also an associate editor from Nature Publishing Journal Clean Water, which is almost 5 impact factor, and many more. So I will not take a lot of time. He was my collaborator at APN project. So Dr. Honda, can you share your experience in next 7 to 10 minutes?
Yeah, thank you, Dr. Kumar. Yeah, I'm happy to meet you here. So, yeah, I don't know what to say, what to talk this session, so I just prepare one slides about, I want to talk about my experience about the COVID-19 surveillance in wastewater. So it's just 5 minutes, I just quickly go. Can I see my slides? Can you see my slides?
Yes, yes, yes. It is full show and it is, yeah.
Okay,
now
so yeah, I'm from Kanazawa University. I've been studying the membrane processes or the antibiotic-resistant bacteria. Now we have studied the SARS-CoV-2 wastewater in wastewater, and I'm from Kanazawa University. Is here. This is Tokyo. This is Osaka. This is the Kanazawa, and this next prefecture. We studied the taking the wastewater sample from the early March. At that time, it was almost really a few cases were reported in these regions. And in the first wave, I think rapid increase started the late March, and the Tokyo was the largest cases per population, and Ishikawa and the Toyama is finally it's become like a second and third largest reported cases per population. So we used the PEC precipitation to concentrate the virus in wastewater, and used the primer probe set for detection of SARS-CoV-2. So we took the samples from the 5 wastewater treatment plants for every week, and this is the results. So this dotted line arrows is negatives, and these are the positive results, and the reds are from the Ishikawa prefecture, the blue are from the Toyama prefecture. So the first detection was in the late March. At that time, there was a reported cases COVID-19. It was the lower than the 1 cases, 0.4 cases per the 100,000 population. And at that time, this is sometimes positive, sometimes negative, probably because of the very low RNA copies, viral copies in the wastewater sample. And as the number of cases increases, the frequency of detection became higher, and the concentration in the wastewater was also become higher. But the estimated RNA concentration in which was just lower than 3 by the 10 to the 4th copies, even at the highest concentration. So in Japan, it was the RNA concentration is much lower than the cases in US or some European countries, probably because of the smaller number of cases. And so in Japan, it is currently the clinical surveillance has many tests. So I don't think it's early warning is not really the good benefit, but limitation clinical surveillance is presence of undiagnosed infections, like about a half of infections are symptomatic, and probably 90% of the infections are mildly symptomatic. So many infected people do not visit clinics. If you do not visit clinics, you will not get a PCR test. You will not diagnosed as a COVID-19. So what we are doing in Japan is every day the 20,000 species tests doing, including the potential the infections. So actually, that's 90% of the tested people are negatives. So actually, we are consuming a lot of money to find the infections and to find the situation of the outbreaks. And even though we tested out like really a lot of people, the accurate species diagnosis is 70%. That means the 30% infection overlooks. So we failed to find that. So the benefits of wastewater-based surveillance is it can test a large group with a small number of tests. It is really cost efficient, and it can capture the infection which are overlooked by clinical surveillance. That means wastewater is collected from the people who do not visit the clinics too. So we can cover all the population in the area. So, and yeah, we also risk when we detect the SARS-CoV-2 in the wastewater. Some people concerned about the risks in the water bodies which receive the treated wastewater. So we we are about, I think our paper is about to publish, almost accepted. So it's really that we review the papers and we don't have the data of the SARS-CoV-2, but we review the papers of knowledge of the envelope viruses or the enteric viruses. So probably the risk of COVID-19 treated wastewater and the receiving water bodies is quite low. It is not really serious, we guess. Okay, so this is another topic. Sorry. So now we in Japan, we organize a COVID-19 task force in the Japan Society of Water Environment as a platform of sharing the up-to-date knowledge on COVID-19 wastewater-based epidemiology and development of SARS-CoV detection methods for wastewater. So we publish tomorrow. We published the SARS-CoV-2 RNA detection manual for wastewater, a premium version of the manual tomorrow, including sampling storage, process controls, control methods, detection methods. So we expect this is used for the local governments or the private sectors in Japan to test the more wastewater for coronavirus. Okay, thank you very much. So we welcome the submission for the Nature Partner Journal Clean Water. So now we have IF, the first year IF, the 4.9, and the next year I heard that there is a positive surprise of IF. So I really encourage you to submit the journal. Thank you.
Yeah, thank you, Professor Honda. Now I would like to, we will get back to you with the panel discussion. So I will hold the question. Of course, the participants can target their question to a specific speaker, and I will take that. So now, Professor Kitajima, I would invite you to share your thoughts, and then Professor Bivinis.
Thank you, Dr. Kumar, for your kind introduction. I'm Masaki Kitajima from Hokkaido University in Japan.
So hold on, Kitajima-san. Yeah. This is my apology.
Okay, please go ahead. Please go ahead.
Actually, this is such a star player. Actually, he looks young. Yesterday, when we were preparing the bio sketchbook, somebody said, "Oh, this is a picture of 1992 or 1998." I don't know. He looks like that. It is our proud that he was our junior at our this one, but his work has been really, really excellent. He superseded many of us, and that's the Professor Kitajima. He has received a lot of best paper award, and I can tell you that when he was studying, at that time itself, he was a very prolific writer. He wanted to publish and many papers in water research before he graduated. He is an International Water Association member, health-related water microbiologist. Willie Grabo Young Investigator Award he received in 2011. He received University of Tokyo President's Award. That is one of the most coveted thing that can be achieved by anybody. And he does have Japan Society of Water Environment recognition. And he is a true virologist. So the floor is yours, Dr. Kitajima.
Okay, thank you very much for your kind introduction again. So I'm Masaki Kitajima from Hokkaido University. So Hokkaido is located in northern part of Japan. So it's snowing outside. It's very cold. Unlike the climate in India. By the way, so today I'd like to talk about some interesting topic related to wastewater surveillance. So the topic I prepared today for this conference is the wastewater-based epidemiology for COVID-19 and Tokyo Olympic Games. So in this presentation, I like to talk about my ongoing efforts on how we can contribute to risk management during the Tokyo Olympic Games as a Japanese scientist. As you may know, we will be holding Tokyo Olympic Games in 2021 summer, next summer in Japan, Tokyo. And it was postponed, unfortunately, due to the global pandemic of COVID-19. So now we will have to organize this big mass gathering event during COVID-19, most likely. With coronavirus crisis. So the risk management of COVID-19 during the Tokyo Olympic Games among the Japanese people, athletes, staff, and audience is the very important task for Japanese scientists. So actually, this is a primary responsibility for relevant Japanese researchers, including myself, I believe. So today I'm going to be talking about how we can contribute to risk management of COVID-19 during Tokyo Olympic Games. As Dr. Honda explained the advantages of wastewater surveillance, we can utilize this surveillance system for more efficient disease surveillance in the population level. So this slide describes how water-based epidemiology can overcome multiple issues of clinical diagnosis. So there are several limitations or issues in clinical diagnosis. Clinical diagnosis is based on mainly PCR tests. For example, there are some limitations of PCR capacity, like due to costs, technicians, PCR facilities, and so on. And as Dr. Honda mentioned earlier, clinical diagnosis basically targets only symptomatic infections. And one of the most significant issues related to COVID-19 is stigma issue for positive individuals, or discrimination for positive individuals. On the other hand, wastewater-based epidemiology can understand the actual infection in the population, including asymptomatic individuals. So we are now trying to establish fundamental technologies to develop early warning and mass diagnosis infrastructure. So this infrastructure is applicable to several applications. The one application is municipal wastewater, like Dr. Honda mentioned in the previous presentation. So through surveillance of wastewater at wastewater treatment plants, we can understand the disease prevalence in the city or town, and so on. And another application is facility-based wastewater surveillance, and that will be my main topic today. So this is the system to understand the facility level of COVID-19 prevalence. And the last application is the wastewater surveillance for mass transportation system, including international flights. So when we test the COVID-19 virus, or I should say SARS-CoV-2, in toilet wastewater from international flights or cruise ships, we can understand infection among immigrants. And this system, facility-based wastewater surveillance and international flight surveillance, is particularly useful to understand the disease prevalence among athletes, staff, and immigrants when we have Tokyo Olympic Games in the next summer. Well, now let me introduce some of my previous studies related to SARS-CoV-2 in wastewater. We wrote a review paper on SARS-CoV-2 in wastewater. Actually, this is the first peer-reviewed review paper proposing the application of COVID-19 wastewater surveillance, wastewater-based epidemiology. We started drafting this manuscript in March, and there was no report on detection of SARS-CoV-2 in wastewater at that time. And this paper was accepted on April 26th, and at that time, only 5 papers, including preprints, were published from all over the world on the detection of SARS-CoV-2 in wastewater. And in this paper, we proposed we highlighted the importance and potential of wastewater-based epidemiology to better understand disease prevalence. And we published press release from my university, Hokkaido University. And then we detected actually SARS-CoV-2 RNA in wastewater in Japan for the first time. We collected wastewater from a wastewater treatment plant located in Yamanashi Prefecture, which is located in the suburbs of Tokyo. The wastewater samples collected were influent secondary treated sewage samples. And in addition to wastewater samples, we collected river water samples as well. And we examined SARS-CoV-2 in those environmental water samples. And we detected SARS-CoV-2 RNA from treated wastewater sample before chlorination, which was collected on April 14th. And actually, this was the period when the number of infected people increased or peaked in this region. So this number is the daily reported cases or cumulative cases in Yamanashi Prefecture. And then we proposed the application of WBE, wastewater-based epidemiology, to aircraft and cruise ship. So I was involved in this study, and one of the panelists, Dr. Bivinis, is also one of the authors of this paper. And we proposed the applicability of wastewater-based epidemiology for mass transportation systems, specifically international flights and cruise ships. Actually, we were able to detect SARS-CoV-2 RNA from toilet wastewater from those systems. Okay, so this is the slide showing the potential applicability of facility level wastewater-based epidemiology, specifically wastewater surveillance in the athlete village of the Tokyo Olympic Games. So this is the expected like structures of the domes or athlete village in Tokyo. And so basically, there are many buildings and facilities, and wastewater will be collected through normal sewer system. So what I'm thinking is we collect wastewater samples from the facilities and then test for COVID-19 and maybe drugs or doping. So we can do COVID-19 testing and doping test for the first time at the same time. So this is basically kill two birds with one stone, I think. But the issue is implementation of this system into Olympic Games athlete village. We are researchers at the university, so we have limited capability for implementation of this technology. We have limited like capacity and testing capacities and labors, manpower. So I'm collaborating with a company in Japan. This Shonogi company is one of the largest pharmaceutical companies in Japan. And we are now actively collaborating with this company to implement wastewater-based epidemiology to many places, including municipal wastewater treatment plants, facility wastewater treatment system, and international flight wastewater surveillance. So we published one article on open like news network, and if you are interested, please look at this. And this is the newest article on this publication. And this is my last slide. So this my idea was conceived by the team called MARCO, Mass Gathering, Risk Control and Communication. This is basically a virtual institute for solution-focused COVID-19 risk assessment. Actually, this virtual institute is not funded yet. So we are using our own money and to implement several technologies to control risks of COVID-19 during mass gathering events. So the most significantly, the Tokyo Olympic Games. So I am in charge of wastewater surveillance in this team, and other team members from diverse disciplines and backgrounds like genomics, public health, medical science, computer science, and so on. So with that, I'd like to thank you for your kind attention, and I will be happy to take any questions in the Q&A session. Thank you very much.
Yeah, thank you. Yeah, this is a summary story, I guess, that how these papers, and of course, I think these things we will, once these are COVID gone, we will be telling these stories nicely. Then now the last speaker for this session, and then we will move to the panel discussion, is Dr. Arun Bivinis. What can I say? I am a little bit jealous about him because he is active in research, he is active on Twitter, he is active on reviewing paper, he is active on every front, and he is active in river catching fish. And according to him is that fish is a big indicator of water quality. And I do agree that just by looking at fish, you can understand the even BOD levels and those things if you have that experience. Dr. Bivinis has emerged as a very great, I think, this global collaboration on COVID, WBE. He was the first author, and where more than 50 institute authors have come along. And I had the privilege to be on that board, and that changed my path. So I would say that a researcher like Bivinis affects a lot of people, and then you know that the other end is also tightly held. He is working at University of Notre Dame with one of the stalwart in this field, that is Dr. Kyle Bibi. And he has the PhD from Georgia Tech. So that is, I think this is, and he is a, you will know that how great he is, just keeping quiet, but once he will start speaking, you will know the energy in this man. Thank you, Dr. Bivinis, for coming on very short notice, and now floor is yours.
Yeah, I'm very happy to join you this morning. I was actually I was a Fulbright Nehru Scholar at CSIR NERI in Nagpur in 2017, working with Dr. Pawan Labhasadwar. And so I'm always very happy to reconnect with colleagues in India and all across the world. So thank you for having me. There is actually a very large Google outage this morning in the United States. So that could explain some of the connection difficulties from this side. I'm actually on my cell phone right now. So anyway, in regards to SARS-CoV-2 and wastewater, yes, there's quite a lot going on, as Dr. Manish Kumar has covered here. And it just so happens that, I guess in February or March, when as this pandemic was beginning to move across the globe, and there were beginning to be reports of this virus in shed and feces, there was a lot of interest in trying to leverage wastewater to manage public health. And so that really what precipitated or caused us to try to write this paper, to kind of try to help pull everyone together, because, you know, the bottom line is that while every individual is brilliant and can go into their lab and I'm sure do the work, that requires that each of us learn the same lessons over and over and over. And so we thought that it would be a great idea to set up an easy way for people to communicate rapidly what they were finding, so that other groups could, you know, move ahead faster. And so we launched a collaborative. There's actually there's a Slack work group that now has, I think, well over 900 researchers listed on the group. And so this is a place where people can share information rapidly. You know, and the idea here is that it's a bit informal, so it's not as long as like a peer review process. So there's obviously some cons associated with that, but the nice thing is it encourages researchers to share their latest findings with other researchers, such that people can adapt quite rapidly. And I think this this group has been key in bringing online several hundred research teams throughout the world. Here in the United States, of course, our case levels are just unbelievable right now. Here in the state of Indiana, we are monitoring at, I'm thinking, so close to 20 wastewater treatment plants here in Indiana. And in 2 of our local plants, you know, as Dr. Kitajima and Dr. Honda were mentioning, one of the key steps here is to concentrate this virus in wastewater. And we've just crossed the threshold here where I'm able to detect it without concentrating the wastewater. So I take a ½ ml of wastewater and able to detect this virus directly, which is quite shocking, to be honest. That just happened last week for the first time. And so it's unfortunately not a good story to tell here in the US. But from a wastewater perspective, it gives us a lot of ability to kind of continue to optimize these methods. And so I guess I'll kind of begin with from the beginning, as Dr. Honda and Kitajima have mentioned. When people are infected with this virus, it infects via a protein that's actually found in the gastrointestinal system. And so we actually see this virus replicating in the intestines and then is shed in feces. And it looks like about anywhere from half to 2/3 of the people infected are going to shed this virus in their feces. And of course, this is collected in a wastewater system in an urban area. And from there, it's, of course, across the population is aggregated. And what's interesting, or one of the difficulties here, is that the shedding density in feces is quite variable, anywhere from 2 log per gram. I've seen as high as 10 log per gram reported for pediatric patients. So we're already, even from step 1, people shedding into the wastewater system, the concentration can vary by 8 orders of magnitude. From here, of course, the feces is collected and it moves through a wastewater collection system, through pipes and pump stations, open channels, whatever the case may be. And of course, there are transport phenomena underway when this virus is moving through these systems. It partitions to solids. It appears to partition to solids preferably over the liquid phase. And then by the time it arrives at the wastewater treatment plant, where most of these measurements are made in the influent, we would expect that there are definitely some decay and partitioning behavior. So we collect a sample from the influent. In most cases, this is a composite sample. So we're collecting a small volume every hour. And then we take across 24 hours, we mix it together, and then we pull one sample from that volume, and we take our measurement for the day. Here we are using the N1 assay, the CDC N1. We found that to be the most sensitive assay. And then also we're using a digital PCR platform, which on our platform, we found that the N1 gene assay appears to perform the best. And so what we've seen here it's important to, I think one of the very important things to recognize here is that there are actually 3 systems here. The first system is of course the clinical surveillance system, which gets a lot of attention. It's the cornerstone of really public health, is the idea that, okay, we can go out and test an individual and determine their disease state. And then there's the wastewater system, which is another surveillance system that also has pros and cons. It's important to remember that the clinical system also has pros and cons, which Dr. Honda and Kitajima have already mentioned. But I think it's also important to recognize that neither of these surveillance systems is the actual truth. That the real truth is a third state. And so when we're comparing clinical surveillance and this wastewater surveillance, it's important to recognize that what we're actually seeing is a difference in the surveillance strategies and what they're able to, their sensitivity in terms of how many cases can we detect, their sensitivity to changes in the transmission patterns in the community. And I think that working together, they both have strengths that we can leverage. But I do want to point that out because I think sometimes people get overly attached to one system where, but even the clinical surveillance is not the truth in the community, it's an approximation. So here in our experience at Notre Dame, we're monitoring in several communities, and what we found is that the advantage that wastewater provides, of course, is the aggregation of all the signal, right? Everyone sheds this virus RNA into the system, and we're able to take a nice sample, a representative sample of that, which allows us to see much of the population. Whereas clinical surveillance, you test one individual, you get information on one individual. But what we found is that depending upon the strength of the clinical surveillance system in that community, wastewater affords more or less advantage in terms of early detection. When the pandemic was first beginning, especially here in the US, we were able to detect RNA pretty soon into that process. I would say that it varies by community. In some communities, there were maybe 50 cases when we detected in wastewater. In other communities, there were more like 100. And that varies based on how the wastewater collection system works, of course. And then we also have industries that are discharging into these systems, which are adding wastewater that contains chemicals that might affect our ability to detect RNA. One of the more interesting things I think here in the US has been that the universities have really taken on a huge amount of this wastewater surveillance as a way of trying to monitor COVID-19 amongst their student populations. And here at Notre Dame, we had a very aggressive clinical testing system. We were doing, I think over the course of the semester, we did almost 90,000 tests, saliva test and swab test. And what we actually found is that the wastewater data, we were collecting wastewater from the outfall of the campus, composites, 24-hour composites daily. And we found that the wastewater data and the clinical data, when the clinical samples were backdated to the date they were collected, that the data actually aligned very, very well with very few days of lead time by the wastewater. But of course, that's in a setting where there are a lot of resources available for clinical testing. 90,000 tests among 13,000 students. So you can do the math on how many times each student was probably tested. When we move into these other communities, municipalities, they don't have the resources to test this aggressively from clinical testing. And so that's where wastewater becomes really useful, because we are able to, at least in 2 local communities, we are able to, twice a week now, we started off daily, but twice a week we collect composite samples, and we of course measure the RNA, and we're able to very clearly distinguish the trends in the infections within the community in this wastewater. And so I'd like to touch a little bit on where we're trying to go with this. Of course, the advantage of our wastewater, if there is that there's potentially up to a week of lead time. 7 to 10 days is what we usually observe over clinical surveillance. And that includes the time required to test the specimen and then get the result back. And so what we're trying to do is we're trying to go faster. And the idea is that, okay, if can I collect a wastewater sample and then have results within a few hours? And so one of the things that we've had some success with and that we're hoping to scale and deploy potentially next semester here at Notre Dame is we're using passive samplers. We're deploying them into the sewers for approximately 4 hours each morning. We then collect them and we squeeze the absorbed sewage out of these samplers. It's a Moore swab. And so we squeeze the sewage out of these Moore swabs. It's usually around 20 mil. We concentrate it using an ultrafilter. We extract it, and then we actually test the extraction using a LAMP kit, which is a loop-mediated isothermal amplification. And so we can go from test to results within about 4 hours. The disadvantage of the LAMP assay is that it's qualitative. It's presence or absence. But it's colorimetric. So what happens is if a sample is positive for the RNA, it turns bright pink. Whereas if it's negative, it stays kind of a yellowish color. And for that reaction to occur, we just have to incubate the sample at, I believe, 56 degrees Celsius for 40 minutes. So it's actually a very streamlined workflow. And so that's kind of what we're interested in here now is trying to produce results faster so that public health agencies can take action faster. So I think that's going to be one of the keys here is, at least here in the US, is delivering results quickly so that public health agencies can take action. So I'm so thrilled to be with you this morning. Thanks for having me. And I'm happy to try and answer any questions that you have in the Q&A.
Yeah, so I mean, you have just finished the panel discussion because the many questions that were burning and I wanted to pose directly, you have answered that. But let me take it through. We have now Professor Bhattacharya also here. He has been also active in this field. And the theme is that COVID-19 and the participation of water scientists. He is one of the great example for that. Let me take this discussion, like we have the three, and if I also count myself, then the four the things. I understand that the media has taken it many times, highlighted it, that some incident, or we found, like you also said, that the wastewater, the results of wastewater aligned with the actual detection date. But at the same time, Dr. Honda has told that asymptomatic patients is a big problem. So what actually we detect? We detect whether or not asymptomatic the patient shed the virus or virus. And if they shed, then when they start shedding, and there is that after some time also it keep on shedding. So what do we detect? Whether that has been said same day. So what is the time interval? That is the I wish to push that and because this is also related to that how much early warning it can be. So I would go with Dr. Honda first, and then yeah. So that yeah.
Okay. So in case of Japan, it's the reported cases and the patients' infections are small. So we didn't see the very effective early warning, probably because of the low concentration virus RNA, and also in the March, during the first outbreak wave, I think many clinics couldn't diagnose the COVID-19. So I think some cities detect the RNA, SARS-CoV-2 RNA earlier in the wastewater, but now in Japan, the hospital clinics is really aggressively the test the people, and they will get back the results very soon. So it's the early warning is kind of the relative relative, I think, things. I mean, the relative. If clinical surveillance get bug result very early, the early warning by the wastewater is not the less effective. But
Yeah.
so I would say so. The benefits of the wastewater surveillance is it may be possible to find the infection which the clinical surveillance overlooked. So now we know the COVID-19, the clinics can diagnose that, but when the another new infectious disease virus occurs, maybe the many clinics cannot diagnose that. At that time, maybe wastewater also works as an early warning system.
Thank you, Dr. Honda. Wow, very interesting answer. I would move to Dr. Kitajima on the same issues.
So, okay, so there are at least two ways of like methods to understand when people start shedding the virus in feces. The first way is like Arun mentioned. Good to see you, Arun, by the way. And so like he said, so we monitor wastewater and we collect clinical samples from individuals, and we compared how long the wastewater data precedes the clinical symptoms of onset and positive tests and so on. And there are some papers, I guess you may know, showing that the increase of SARS-CoV-2 RNA concentration in wastewater or wastewater sludge precedes the increase of the number of patients or number of positive individuals in the same region. So this is like population-based surveillance data, right? On the other hand, we can also track the individual tests as well. But the clinical surveillance, current clinical surveillance cannot go back to the dates before the patients exhibit symptoms, right? So now we cannot understand how early they start shedding virus in feces. So this is a limitation of our current clinical data. Actually, we just published as a preprint paper just now, a couple weeks ago. In this paper, we analyzed clinical shedding patterns over time. So according to our analysis, so right after exhibiting symptoms, fecal viral concentration already peaked in the immediately after. So immediately after means within 1 day after exhibiting symptoms. This indicates that people started shedding viruses before they exhibit clinical symptoms. So this is our suggestion from our analysis of quantitative data from shedding virus from individual. And so way forward, now clinical testing or clinical challenge tests for vaccine trial is ongoing, especially in the UK. So we will have like prospective data from clinical testing and vaccine challenge studies. So from this data, probably we will have to, or we'll be able to obtain very clear data on how early they start shedding virus in feces before exhibiting symptoms. So yeah, and another thing, so there are some limitations for early warning systems of WBE, wastewater surveillance, to be honest. Although I'm working on wastewater surveillance.
No, there is nothing bad. I think every one of us has accepted that, and we have already told that. Only thing is that what I want participants and myself, and also like some of the key matter already you both have pointed out, that this can give you a chance to go back into the history of a patient. And like Dr. Honda has told, that maybe this time But if we can keep this surveillance going, next time we will be at first, and we can say that the wastewater surveillance has many positive things that has been recognized by no doubt. However, like if there will be not consensus among the worker, that among a worker like us, that how much early warning it can give. Like just now, Honda Sensei has told that this is not much lag behind. My result says that a week, or sometimes because we don't have a lot of good data of the catchment area, within the catchment area, because we have the entire city data. But we don't have that particular wastewater treatment is serving how many people. So in that sense, still I can find that a week to 2 weeks max, but that is the early warning. And of course we can see some relationship. So coming to that is that that's why that question was important, that what we are determining actually, and then how much early warning it can give. So I would invite Dr. Bivinis to let us understand or give that okay, although he has talked about several things, but how many population and gene correlation? I mean, do we have any coefficient that, because we know that infection stage can determine how much copies of a gene can be excreted by an individual. So whether the increase or decrease, how we can compensate with that data, and how much early warning we can get. And yeah, this is these two, and then I will pose the last round of the question, and then we'll end up.
Yeah, no, I think this is these are pretty much at the center of the use case for wastewater-based epidemiology. I think the point I would like to make in all of this is I'm about 6'3" tall, so that's 190 centimeters. And you know, if I stand next to someone that's 150 centimeters tall, I look very tall. But if I go and stand next to LeBron James, I don't look so tall anymore. In fact, I look short. And I think when we consider the potential of wastewater-based epidemiology, we have to look at the context. You know, here at Notre Dame, I can go take a clinical test, a saliva test, actually. You know, you have to spit into a tube and they test it, and I get the results back in less than 24 hours. It's usually anywhere from 14 to 16 hours. And so that's a very fast turnaround for that information. However, if I drive my car about 30 miles to the municipality in the next county over, and I go in for a clinical test, it could be 2 weeks before I get my result back. And so those are two very different contexts, and wastewater can add probably a lot of value in the context where it's a 2-week period before I know my clinical results. Because the wastewater, you know, I'd say realistically, we can turn around, if we really want to go as fast as we can turn around, you know, results in 24 hours for probably 16 samples every 24 hours. So, you know, when we interpret the capability of this approach, we have to consider the context. And in contexts where clinical symptoms are very short, wastewater-based epidemiology is going to look quite tall. But in situations like where clinical testing is easily available with a rapid turnaround, we're probably not going to see as much advantage in terms of lead time, early warning, but there's still advantage in terms of monitoring asymptomatic or pre-symptomatic infections. And I think it's kind of important to recognize that pre-symptomatic and asymptomatic infections are usually where we have limited amounts of data, because these folks are not yet showing any symptoms, and so we don't know to be measuring anything from them. So yeah, I think it's going to vary a lot by context, what wastewater-based epidemiology can offer. There has been a paper, I think it might still be in preprint form, from Stanford. And what they found was that if you compare the date the result was reported, then wastewater leads infection by clinical by 7 days. But if you take those clinical results and you backdate them to the date that the specimen was actually collected, then wastewater only leads by 0 to 2 days. And to be honest, I think that 0 to 2 days more closely reflects the biological system, what's actually biologically happening in an individual, versus the 7-day to 10-day lead time represents differences in the surveillance systems themselves, not biological phenomena within an infected individual. There's actually a lot of really nice papers on African green monkeys have been inoculated with SARS-CoV-2, and then their shedding has been monitored and longitudinal. And there's some really nice literature there in terms of, from day of inoculation. It seems like within probably 3 to 4 days, the shedding and feces peaks. And that peak is strongly correlated with the peak in the nasopharyngeal swabs as well. So that's a good starting point. And as Dr. Kitajima mentioned, hopefully soon we'll have some better data in humans.
Actually, when you speak, I just want to keep hearing because we professors like that try to learn from each other. So this is because I will take only two more questions, and then we will end up this session, because next speaker, Professor David Warner, and my co-convener is here now. So community, yeah, you can say hi to everybody. They have really come at very short notice, and we can only be thankful for that. There is the two things. One is about size of the treatment plants, and then there is one argument about that in a city, how many surveillance has to be done. I mean, if I want to monitor Ahmedabad city or Gandhinagar, how many samples should I collect? Not that in a given wastewater treatment plant, how many samples I should take in a day. But if I always go to a water manager or policy maker, they say that, okay, tell me how many sampling will be sufficient. And I don't have a lot of a thumb rule. I try to search Bibi et al. Or Kitajima et al., but I didn't find anything in that sense, that how many samples. Then whether, if we do not monitor wastewater, can we, how about the drain system? Can drain system lead to like main sewer system? Can we put it at a, like the network, if we go and sample at a major point, connecting point, can we tell the variation in the city? So about that, sampling points, how many sampling points and where to sample? Anybody can go ahead.
Well, I think, just to get the conversation started, you have to decide what your objective is first. Are you trying to detect hotspots to follow up with clinical surveillance? In that case, there probably needs to be more sampling points, and you probably need to push them up into the sewer system so that you can actually identify geographic areas for follow-up clinical testing. If you're just trying to measure infection trends within a city, then you can probably focus on more centralized treatment locations. And in regards to open drains, there's actually teams that have been doing that in Bangladesh. There's also, my understanding is Andre Badia, who's in Costa Rica, his team has been monitoring open drains using more swabs. Anywhere where this fecal signal is likely to go or likely to be is a worthy monitoring point. But I would say that if you're just interested in monitoring trends over time, you can probably get away with fewer monitoring points at centralized locations. If you want to target clinical testing to interrupt transmission chains, you probably need more samples, and those samples should be collected from collectors that would allow you to identify specific areas for clinical testing.
Okay, so the next person, anybody wants to add to this? Of course, the Arun is taking all this and explanatory. So
yeah, so I agree with Arun's comments. So yeah, it depends on the purpose, your purpose. Yeah, like Arun mentioned, we could collect from sewer networks, from like manholes or from facility like septic tanks or wastewater treatment plants. But it really depends on what your purpose is. So and if you collect wastewater from municipal wastewater treatment plants, diurnal or within day variation is relatively low. But if you collect from upstream of this network, like manholes or facility, the daily variation or within day variation is very high. So as far as I have investigated so far, I've been collecting wastewater samples from a hospital, and the fecal load or fecal viral load varies more than 1 log within a day. So from facility level wastewater, which means that depending on the time you collect wastewater samples, the sensitivity differs by 1 log. So this is the observation that I have observed, as you may imagine from. So the sampling locations and when you collect and what type of samples, grab samples or composite samples, it really depends on where you collected samples from and what the purpose is.
Great. Honda-san, any your?
Yeah, most are already pointed by Daro and Masaki, but yeah, I would say so. Yeah, smaller catchments usually has a larger fluctuation, so probably composite sample is more, I think, more chance to catch the. Oh, sorry, can I? Yeah, hello.
Yeah, I can hear you.
Yeah, more chance to catch the infection, and if the reported number of patients is small, so if we, for example, there are only 1 patient in the catchments, so we have to take the wastewater sample when he or she goes to the toilet, right? So at that time, it is hard to catch the infection by the grab sampling. But if there's a certain number of group of infections and the catchments are larger, maybe you can take by the grab sampling. And if there's a kind of peak of fecal discharge, maybe you can detect more sensitive. I think you can get a higher peak than the composite sample. So yeah, I think it also depends on the situations of the infections and your target catchment.
Yeah, so I told that I have only 2 questions. Which means that I should be ethically should be on the last question, but because you just talked about this word infectivity, I just want to take a very short, maybe 10-second answer from you all. Is that how much do you think is that these genes, that is maybe the part of it, genome and the RNA, how much of them are infected? Because before going to that you try on answering, I would suggest that I understand that this virus cannot bear our stomach or the digestive juices. So if it is coming through the fecal matter, of course, there is no whatsoever infectivity remains. However, if it comes with the sputum or the swab or the saliva while washing mouth, they are definitely going to be infected. So what do you think is the infection level? Because now little bit people are ready, because earlier we were not trying to have that, okay, this is infective, it would have been a very havoc. So I want to listen from you 3 guys, that what do you think is the probability of infection through this wastewater?
Okay, so let me initiate the discussion. Okay, so in the first place, from fecal samples, there is no quantitative data on the infective viral titer in feces. So this is one point. We have a lot of RNA copy data from feces, like Arun mentioned, but for infectivity data, it's very limited. And according to Arun's paper, so the ratio between infective viral particles and the total number of viral RNA copies in water changes over time. So the ratio changes over time, which means infectivity decreases more rapidly than RNA copy numbers. So RNA copies stays much longer in water. But the degradation largely depends on water temperature. Yeah, so this is a very important paper published by Arun. So yeah, this is my comment. And yeah,
thank you for being very crisp and to the point.
Yeah, I think that's right on. I think the main thing is to recognize that the risk is never zero, but just because something is possible does not mean it's probable. And so it's very, very important that we're working in resource scarce conditions, right? We only have so much PPE, we only have so much resources that we can use in fighting against this pandemic, and so we have to do triage. And so I would say that while possible, I think on the list of things we should be worried about, that one's farther down the list, in my opinion.
Great. Honda-san?
Yeah, I think this is very important, but risk is not zero, and it also depends on the situation. And I would say there's a report in China that's in the area which has a low sanitation, there's a possible transmission from the sewage. It's I think the toilet pipeline probably. So it is possible, and it is hard. We don't have the currently it is hard to calculate the risk in the quantitatively, but the qualitatively, if the wastewater comes from the close to the source, it has a higher risk. And in the area, if there's more larger number of infection, there is more risk. There will be a high risk. So if far from the source and less infection, there will be a risk lower. But we should not consider it with a zero.
Yeah, I remember you saying. I mean, actually, I and Honda-san. Yeah. So I mean, I quote many times when he says that, see, proving something not there is almost proving something there is easy, but proving something not there it's so kind of impossible. So I think, but he uses different information today, but the same point. Last but the not least, what is the situation in your countries? Because whatever the wastewater surveillance system goes, but in India, sadly, researchers are trying to do it, but government has not taken it up in a big way, or even in an average or some way. So I want to know the condition in your countries. Of course, Arun has told about the campus. My campus has taken up. They have immediately given me grant from this KPCSD, and I am observing the entire city, including the Gandhinagar. And probably within a week or so, our paper will be out. So, but what is the general consensus about the city?
Okay, so let me start. Can I?
Yeah, please.
Okay, so I assume Dr. Honda will talk about situation in Japan with respect to municipal wastewater surveillance. But let me talk about facility level wastewater surveillance in my country that is aligned with my topic today. So, okay, so you know pooling tests in clinical diagnosis, right? So pooling test means you mix clinical specimens from multiple individuals. If the pooled sample is tested negative, then you consider all tested individuals are negative for the pooled specimens. So I think the wastewater surveillance is the analogy of pooling tests for the clinical diagnosis. What I mean is, negative results from wastewater surveillance is also important in addition to positive results and quantitative data, which means that by testing wastewater samples, which was collected like composite samples, 24-hour composite samples, which represents the population, then we test SARS-CoV-2, and if the virus is negative, then we consider maybe there is no infections or a very low number of infections in the community. So this is the analogy of pooling tests. So this is another way of the application of this test. So especially for Olympic Games, athletes not expected to be infected with COVID-19, right? So it is very important to confirm that wastewater is negative from for the Olympic athletes village. This is just one example, but this is another way of thinking of application of WBE for COVID-19 surveillance. Thanks.
Wow, very nice. Honda-san.
Yeah, in Japan, I think some cities interested in, some cities not. But I think it is not on their hands, but our hands. That means that I think they are not. They don't know. We don't know how reliable the information of the COVID-19 and the SARS-CoV-2 RNA wastewater, and it's the quality of that, the certainty or the accuracy of that. So we should show that. In our case, we don't have much data on that yet. So we have to accumulate data, and we have to show the detection or negative results shows what kind of conditions, situations. It is really important. So I think clinical surveillance is always a primary indicator of the to know the pandemic situation, and I think the wastewater surveillance is the information to complement that. So early warning is the one of the complement, and also if sometimes there's no reported cases by clinical surveillance, but the SARS-CoV-2 RNA is taken in the wastewater. So that is some people are infected but undiagnosed. So
yeah, actually this is very, very interesting point. Actually, in my case also, I have seen that it is positive, but no cases reported.
Yeah, so we can say sometimes like this, sometimes this case was this, but we need more reliable. I mean, the more.
So, I mean, somehow the Japanese municipal corporation or cities are not also. I mean, a kind of Indian situation is there.
Yeah, yes, they are. I think. Yes, they are. I think waiting. I think they are. They are not still. Yeah, still step back and see what is happening.
Yeah,
is it really reliable or not? Is it useful or not? So I think it's in our hands. So in case of Japan, we cannot correlate the viral titer to the number of reported cases. So it's really small number of cases. So probably it's really high fluctuated when the reported case is small. So what is possible, what it was in Japan in current situation is that when we detect the SARS-CoV-2 RNA in wastewater, there will be some patients in the catchment. But the important point is that when we didn't detect the RNA titer, how much is the probability there is no infected people? Is it 50 or 80%? That is really important. So we have to have some quantitative and quality management.
Yeah, actually, let's ask from David. David, what is the situation in UK? Although it seems like they have included it in national surveillance. And actually, then we will go back to maybe Professor Bhattacharya for knowing about another European case, and then ends with Arun. And then we will wrap up this the thing. So, David, what is the situation in UK?
Yeah, hello, everyone. And really nice and interesting discussions here that I've been able to listen into. And apologies for being slightly late, but I had to teach online. So, I mean, I'm not doing COVID work myself, but my colleague does, and he does this as part of a national surveillance program. So there is a national effort to survey COVID-19 in wastewater in the UK that's currently monitoring, I'm not sure how many, but at least 10 different sites in different parts of the country. My understanding from how well that's going is that there's still a lot of uncertainty around how to measure actually and reliably measure your gene copies. And a lot of the results are also very close to the detection limit. So I think, you know, there's still a lot of science that needs to be done, but there are signs that this can be valuable. And in the UK, it's something that the government is investing in.
Yeah, thank you. So Professor Bhattacharya, in short, what is the situation in Europe? You have to unmute yourself.
Thank you very much, and thanks to Arun, Ryo, and also Masaki. I've heard so much about you. Good to see you live, and your comments on diverse issues on COVID. But the major question which I would like to ask right now, because in the whole process started from the Netherlands during the very early stage of the COVID disease outbreak, and that was from February through April, where they have sequentially looked into the progress of the pandemic. I would say progress of the pandemic because they were monitoring simultaneously during that, and there were a clear indication of how the number of gene copies were increasing with the increase in the outbreaks, right? Except there in Netherlands, it was not a major problem. Here, even in Sweden, we could also see especially the catchments where we had exceedingly high disease burden. Those specific inlets were having high number of gene copies to start with. Now, my question, which I've just posted out to all panelists here, as it is now, it's a point of debate, definitely, but there will be also research. But how do we really see these indicative numbers, indicative figures, to at least look into the problem? Because right now, what we are doing as scientists, we are just beating the bush because we are not processing or proceeding in a very scientific manner. Lockdown and not case sensitive lockdowns and non-case sensitive lockdowns. It is just presumably fully based on assumptions. And even we are working from home, we don't know what is the extent of the disease transmissions, even if you're at a job or traveling, so on and so forth. Now, the question is, if you can really see the total number of virus counts and try to build up a benchmark, yes, what can we optimize before we refine it? Because even all scientific theories, they're subject to refinements. But we need to start at some point where we can say that concretely, say, yes, these are the current numbers. However, we can really enhance the sensitivity of our RNA separation techniques. So I mean, we need whatever we say, we need to say with a bit high level of confidence so that the policymakers don't get confused. So that is we are not yet there. And that is one of the main reasons why in Swedish environment, this national, the agency, they have been quite skeptical, because if we do not convey our results in a more convincing manner, then nobody else, if we are not convinced, nobody else will be convinced. So that's the status. And I think I would stop there, but with an urge that let's not be shy about our data, but we keep on that. Yes, if we see the direct increase in the our number of genomic copies in wastewater, so we can put some sort of threshold at the point. That's what I mean. Thank you.
So Arun, now because you were giving the last, last to speak. Now you can be the last to finish. So then, with your comment, we will finish. So please tell me that how do you? What is the situation in the US, and what you see we are moving to?
Yeah,
and how can
I
we
think
convince these people that to adopt? Because I see that Kitajima is saying that at least it will tell you that whatever the secure place is, unless what time it is secure. That is one of the very key. And there are several other, Dr. Honda has already told that at least it can give us something is there or something is not there. So your say.
Yeah, I think here in the US, it seems like most of the effort has been led by university research teams, and it's been specific to college campuses. But what's nice about those is they kind of are really nice case studies because it's a small, you know, it's a most campuses in the US are like miniature cities. And so there are a lot of university groups leading this, but also now the federal government has awarded a contract for monitoring from, I'm not sure how many cities, but it's supposedly, I think, going to be several, up to half of the US population will be covered, will be contributing wastewater to the cities that are monitored. So the federal government has put some money in. The other thing I would point out is Biobot Analytics, which is a startup company out of Massachusetts. They had a fundraising round at the beginning of this pandemic, and they were able to raise, I think, $6.5 million. So it seems that, you know, the government is moving towards trying to scale up this approach, but also the businesses seem, you know, investors seem willing to take a risk on this approach and its potential. And I think that, you know, in my mind, as Dr. Kitajima mentioned, one of the strongest use cases for this is facility level monitoring, because it's very specific to who's in that building, so that if we actually do get a detection, we can do something about it. And, you know, I think that we can, if we can come up with methods that are affordable, accessible, and rapid, then we can probably offer some reasonable approaches there. One thing around methods, you know, there's been a lot of effort here in North America, Canada, and the United States to come up with a standard method. And all these methods comparisons seem to wind up in the same place, which is that the variability between them is, the variability within each method is such that there's really no difference in the in the methods. And so, you know, I'm not sure how close we're ever going to get to a standard approach. But, you know, here in the US, unfortunately, our caseload is incredibly high. And so you know, I think the use case for wastewater is actually quite a bit stronger because of that.
Thank you so much. Actually, I feel like you people are right there who is heading this branch of science. And with the constant publications that just now Honda-san has come out, the Masaki Kitajima is like the most of the publications were to say that, look, it has the potential, and it is saying again and again. And if now my contribution is coming, let's see that we will be able to convince them. UK, I think it says about the climate change also that sometimes it is better to be on the safer side rather than let the technique be proved very soundproof and like that. So it was amazing to hear you. So I can only say that I could have put 3 days COVID the conference. But thank you very much. You guys just saved me by because this project was given an extension until January 5th. And so suddenly we were in a, we wanted to do that, and you came up, you gave your best, and you all were in the best form of yourself. I enjoyed thoroughly. Thank you so much. If you wish to stay, please stay, because we are moving to the next session. And if you want to leave, of course, you can leave. But a token of thanks will be sent. And of course, I am always there if you want to use me anywhere. So thank you very much for being our panelist. Thank you very much.
Thank you very much.
Thank you.
Thank you.
Bye.
Okay, then.